Dear friends, once again hearty and warm welcome. Friends, in this our new lecture we are going to learn new topic, that is DNA packaging. DNA packaging is considered as one of the most important activity, one of the most important process in the cells of living organism. First of all, let me tell you the basic definition of DNA packaging. DNA packaging म्हणजे काय याची एक सर्वसाधारण व्याख्या या ठिकाणी आधी मी तुमच्या समोर मांडतोय. The DNA packaging is defined as it is the systematic arrangement of DNA. It is the systematic arrangement of DNA on the protein core within the nucleus of living cell. This is the basic definition of DNA packaging. I am going to repeat once again. DNA packaging is defined as it is the systematic arrangement of DNA on the protein core within the nucleus of cells of living organism. It means that the DNA packaging involves the accommodation of highly elongated and large amount of DNA within a very small sized nucleus. DNA packaging, it involves accommodation or packing, tight packing of highly elongated DNA molecules and large amount of DNA within a very small sized nucleus. I would like to give an example. Say, in mammalian cell, the approximate size of nucleus is 10 raised to minus 4 meter. Listen, I said that in the nucleus of mammalian cell, nucleus of mammalian cell has approximate size of 10 raised to minus 4 meter, and the average size of DNA molecule, average size of DNA molecule is about 2.2 meter. So obviously such highly elongated DNA molecule, it can be properly accommodated, it can be properly packed only because of its condensation, only because of coiling and supercoiling of the DNA molecule, and simply this process in which such large amount of DNA and highly elongated DNA molecules undergo condensation, that is coiling, supercoiling, so that it can be easily packed, accommodated within a small sized nucleus, is known as DNA packaging. I would like to give an example. Consider, suppose here I have a thread. Here I have a thread, very much elongated thread, you can see. And here I have a matchbox. Now consider this matchbox as a nucleus of cell, nucleus of any living cell, and this highly elongated thread is nothing but the DNA. Now if we want to pack, if we want to adjust, accommodated such an elongated thread within such a small sized matchbox, then definitely its coiling, supercoiling is necessary. So this DNA molecule undergoes coiling, supercoiling, that is condensation, so that it can be easily packed within a small sized nucleus, and this process is simply called as the DNA packaging. Now we people have to study the process of DNA packaging in prokaryotic organism, that is prokaryotes, as well as DNA packaging in eukaryotic organism, that is eukaryotes, that is eukaryotic cells. So first of all, let us talk about DNA packaging in prokaryotes. packaging of DNA in prokaryotes. In prokaryotic organisms, we know the nucleus is undeveloped. Prokaryotic, that is, pro stands for undeveloped and karyon stands for the nucleus. So in prokaryotic organisms such as bacteria, cyanobacteria, cyanobacteria are also called as the BGA, that is blue-green algae. So in prokaryotes like bacteria, cyanobacteria, in blue-green algae, well-developed nucleus is absent and the genetic material is represented by single, circular, double-stranded and highly condensed DNA molecule. Am I right? I said that in prokaryotic organisms like bacteria and cyanobacteria, the nucleus is undeveloped and hence the genetic material is represented by single, circular, double-stranded and highly condensed DNA molecule, which may be known as bacterial chromosome, bacterial DNA, or simply known as nucleoid. Let me draw here a diagram. Consider here is supposed to be the single, circular, double-stranded and highly condensed DNA molecule. In this diagram, I have shown the relaxed, fully relaxed or uncondensed DNA molecule. We know it is double-stranded, though in diagram I have shown only a single line. Now, the diameter of such single circular bacterial DNA is near about 350 micron. 350 micron. Consider it is supposed to be the fully relaxed or uncondensed bacterial DNA. Bacterial chromosome. I said it is also known as nucleoid. Okay. And the size of this bacterial DNA is near about 1100 micron in perimeter. It means when this circular DNA is cut and stretched out completely, its size is near about 1100 micron, while the diameter of circular DNA is about 350 micron. On the other hand, the size of bacterial cell, consider E. coli. The size of bacterial cell E. coli is simply 2 to 3 micron. 2 to 3 micron. And therefore, for the proper accommodation of such large sized DNA, the condensation, coiling, supercoiling, that is packaging of this bacterial DNA is necessary. Now this circular DNA, it shows looping. Circular DNA, it shows looping, means it forms about 40 to 50 folds upon itself. 40 to 50 folds upon itself. Though in diagram I am going to show only few loops, but actually, in fact, this circular DNA shows about 40 to 50 loops. 40 to 50 folds. These loops are also called as looped domains. Looped domains. And these looped domains are held together by means of some RNA molecules. By means of some RNA molecules. Look here in the diagram. These RNA molecules are also known as RNA connectors. RNA connectors. So, the circular DNA shows looping, and because of looping, because of formation of such 40 to 50 looped domains, the size of this bacterial chromosome is reduced to near about 30 micron. You know, the original diameter of bacterial DNA is about 350 micron, but because of formation of such looped domains, now its size get reduced to about 30 micron. Now each DNA segment of each looped domain, the DNA segment of each looped domain further shows coiling and supercoiling. Further shows coiling and supercoiling. Here in diagram I am going to show. Look, the DNA segment of each looped domain further shows such type of coiling and supercoiling, because of which again the size of bacterial chromosome get reduced to only 2 micron. Here we can say it is supercoiled bacterial DNA, supercoiled nucleoid with diameter of 2 micron, and here this supercoiled or highly condensed structure of nucleoid is facilitated, is assisted with RNA connectors as well as few non-histone proteins. Few non-histone proteins. Mind well, in prokaryotic cells, histone proteins are absent. So, such highly condensed structure of nucleoid is assisted by RNA connectors, non-histone proteins which are, say, positively charged. Non-histone proteins are positively charged. We know the DNA is negatively charged, and these positively charged non-histone proteins along with some enzymes such as DNA gyrase and DNA topoisomerase first maintain such highly condensed structure of DNA nucleoid. So, because of looping, because of coiling and supercoiling, that is condensation, such highly elongated and large sized bacterial DNA can be easily accommodated within a small sized bacterial cell, and this is only because of DNA packaging. This is the advantage of DNA packaging. So here we have studied the mechanism or process of DNA packaging in prokaryotes. Now let us talk about DNA packaging in eukaryotes. Packaging of DNA in eukaryotes. Just now we have studied about the DNA packaging in prokaryotes, and here let us talk about DNA packaging in eukaryotes. We know eukaryotes, that is the cells with well developed nucleus. Now in eukaryotic organisms and eukaryotic cells, the nucleus is advanced, well developed, and shows different components such as, let me draw here a small diagram of eukaryotic nucleus. The eukaryotic nucleus consists of different components such as the nuclear membrane, single or more nucleoli. Here I have shown only single nucleolus. Let me label. Here is single nucleolus. Nuclear membrane, it encloses a viscous material known as nucleoplasm, and nucleoplasm contain many very thin elongated network of chromosomal fibers. Network of chromosomal fibers. You know, these chromosomal fibers are made up of nucleoproteins. That is, chromosomal fibers are made up of nucleic acid, DNA and histone proteins. Histone proteins, and thus these chromosomal fibers are also known as chromatin fibers. And the network of these chromatin fibers in the nucleoplasm of eukaryotic nucleus is also known as chromatin network. Okay. Now, the chromatin fibers, I said just now, made up of DNA and histone protein. Under electron microscope, each chromatin fiber appears just like a string of beads. Under electron microscope, chromatin fiber appears just like a string of beads. Let me explain you. Suppose here is a single chromatin fiber. I am going to show these bead-like structures. Bead in the sense small rounded bodies. And here is a thread by which all these beads are interconnected with each other. So I said that under electron microscope, each chromatin fiber, it appears just like a string of beads. I have prepared one very small model. You can see. Simply here I have prepared a very simple model in which here many small pieces of chalks are held together with the help of a thread. So simply the chromatin fiber under electron microscope, it looks like this. That is the string of beads. Now this beaded string, that is the string of beads, shows many repeating units. You can see with the help of yellow chalk I have shown. This string of bead, this beaded string shows many repeating units, that is beads, known as nucleosome. Known as nucleosome. Each bead-like structure is known as nucleosome. Now this nucleosome is made up of two components. Let me write here as central core and DNA segment. Central core and DNA segment. The central core is a bead-like structure and is made up of two molecules of each of four different types of histone proteins. Listen, this central core is made up of two molecules of each of four different types of histone proteins. Four different types of histone proteins such as H2A, H2B, H3 and H4. These are four different types of histone proteins, and two molecules of each of these four histone proteins togetherly form a group of 8 histone protein molecules. Hence it is also known as Histone Octomer. Central core may be known as Histone Octomer or may be called as octamer core or may be called as new body. New body. These are the different names. Diagrammatically, let me show you. Please look at the diagram. Consider here are two molecules of H2A. Here is H2A, H2A. Here are two molecules of H2B. Here are two molecules of H3. Just consider H3 and two molecules of H4. You must have understanding. So I said that the two molecules of each of these four histone proteins togetherly form a group, a mass known as histone octomer, or it is known as the central core. Basically, these all histone proteins are very rich in basic amino acids, namely lysine and arginine, and these have positive charge along their side chain. Now these histone proteins are positively charged and DNA is negatively charged, and thus the negatively charged DNA molecule is tightly wrapped, tightly coiled around the histone octomer. Let me explain you. Now here we have seen this is nothing but the histone octomer. The DNA molecule, it takes 1 and 3/4 coil around the histone octomer. Here in diagram I have shown with the help of red chalk the DNA molecule, and I said the DNA, it takes 1 and 3/4 coil, 1 and 3/4 coil around the histone octomer. This DNA segment which coils around the histone octomer is known as core DNA. It is known as core DNA. Very simple. Because this DNA segment, it is wrapped, it coils around the central core, and therefore it is known as the core DNA. And this core DNA consists of 146 base pairs. You know DNA, it is polynucleotide molecule. In 11th we have studied, and the double stranded DNA shows the thousands of base pairs, isn't it? Adenine, thymine, guanine, cytosine. So here the core DNA, it consists of 146 base pairs, while a short segment of DNA which joins two successive histone octomers, a short segment of DNA which joins two successive histone octomers is known as linker DNA. Let me explain you. With the help of yellow chalk, here I have shown one histone octomer, okay, and here I am going to show another histone octomer. Look. Consider here is another histone octomer. So I said that a short segment of DNA which connect two successive histone octomers is known as linker DNA, and it consists of 54 base pairs, and that linker DNA further associated with H1 histone protein molecule, single molecule of H1 histone protein. Let me show. Suppose here is H1 histone protein. H1 histone protein. It remains associated to the linker DNA. Now the diameter of this histone octomer is 100 angstrom. It is 100 angstrom. Try to understand. That is nothing but the 10 nanometer. 100 angstrom or 10 nanometer. And such thin elongated string of beads undergoes coiling to form nucleosome fiber having the diameter of about 10 nanometer or 100 angstrom. 100 angstrom. Now here this nucleosome fiber does not show the linker DNA. Please, I want to correct. In this nucleosome fiber, the linker DNA is not visible. Linker DNA is not visible. Try to understand. Here in this string of beads, the linker DNA is visible, but as it undergoes coiling, therefore now the linker DNA is not visible in nucleosome fiber. Nucleosome fiber is the characteristic feature of S phase, of interphase, of cell cycle. It is the characteristic feature of S phase. You know S phase, synthetic phase, of interphase, of cell cycle, we people have studied in 11. Okay. Now the nucleosome fiber, it further undergoes coiling, supercoiling when it starts the cell cycle. When it starts the first phase of karyokinesis of cell division, that is prophase. Nucleosome fiber further undergoes coiling and supercoiling to form solenoid fiber having the thickness or diameter of about 30 nanometer or 300 angstrom. 300 angstrom. Now this nucleosome fiber and this solenoid fiber are the characteristic of interphasic nucleus. Characteristic of interphasic nucleus. And as it starts the process of cell division during prophase, the solenoid fiber further undergoes coiling, supercoiling, that is condensation, to form the chromatin fiber of thickness 200 nanometer. Try to understand. 200 nanometer. And in metaphase, this chromatin fiber further undergoes condensation so as to form a thick and short chromatids having the thickness of about 700 nanometer. 700 nanometer. We know that in metaphase, the sister chromatids of each chromosome are held together with the help of common structure, that is nothing but the centromere. In this manner, due to such coiling and supercoiling shown by these chromatin fibers or chromosomal fibers, that is the string of beads, highly elongated DNA molecules, they can be easily condensed within a small sized nucleus. I would like to tell you, for the higher level of packaging of chromatin fibers, it needs certain non-histone chromosomal proteins also. Non-histone chromosomal proteins also. In short, non-histone chromosomal proteins, NHC. So, for higher level of packaging of DNA, for higher level of condensation of DNA, these non-histone chromosomal proteins are also necessary. These non-histone chromosomal proteins does not only assist for the DNA packaging, but it also stimulates the genetic activity. So here we have considered and studied about the packaging of DNA in eukaryotes. Now let us talk briefly about the types of chromatin material. There are two types, such as euchromatin and heterochromatin. Let me explain you. Types of chromatin material. Basically there are two types, such as euchromatin and second heterochromatin. Euchromatin and heterochromatin. Euchromatin means a loosely packed region of chromatin of the chromosome or chromonema and stands light, that is receives less stain, is known as euchromatin. I said that a loosely packed, loosely condensed region of chromatin of the chromosome or chromonema and which is less stain, that is which receives less stain, and that's why its stain light is known as euchromatin. Let me explain you. While studying the chromosome, we have seen that there are presence of chromonema, such type of chromonema which lie parallel to each other. Now this chromonema consists of chromatin material and the region of chromatin material which is loosely packed. Let me show. Suppose this is loosely packed region. Consider. And this loosely packed region of chromatin which stain less or light is known as euchromatin. This euchromatin is metabolically active region or transcriptionally active region. What does it mean? You will understand later on in the same chapter. But temporarily you just remember that euchromatin is metabolically or transcriptionally active region and it undergoes replication earlier during the S phase of interphase of cell cycle. याचा अर्थ क्रोमॅटिन मटेरियलचा जो भाग हा लूजली पॅक्ड आहे, जो कमी प्रमाणात स्टेन रिसीव्ह करतो आणि त्यामुळे तो लाईट अपीयर होतो, तसंच हा ऍक्टिव्ह पार्ट मानला जातो की जो ट्रान्सक्रिप्शन या क्रियेमध्ये सहभागी होतो. एवढंच नाही तर ज्या वेळेला डीएनएचं रेप्लिकेशन होतं त्यावेळेला या यूक्रोमॅटिनचं फास्ट आणि आधी रेप्लिकेशन होत असतं. या उलट द हेटेरोक्रोमॅटिन मीन्स इट इज द कंडेन्स्ड region. You just compare. This is supposed to be the loosely packed region and it is condensed region. This condensed region is known as heterochromatin. Tightly packed region, condensed or packed region, and it stains dark, means it receives heavy stain, it is deeply stained. This region is known as heterochromatin. Heterochromatin is considered as metabolically or transcriptionally inactive part. Also, it undergoes replication slowly and late in the interphase of cell cycle. So here we have considered about the types of chromatin, and before to that we have finished the topic, that is the DNA packaging. So let us stop here. Thank you.